In this lecture, we're going to take a look at the processes of transcription and translation. The nucleic acid called RNA is known to be the bridge between the DNA of our genes and the proteins for which they code. Transcription is defined as the synthesis of an RNA transcript from a DNA template. It uses the genetic information that is contained within the DNA sequences that we call genes. And once that RNA has been transcribed, it undergoes a process called translation. Translation is the synthesis of a polypeptide chain from an RNA message. It uses the information encoded in that RNA message to result in a unique combination of amino acids that we call a polypeptide chain. That polypeptide chain is then folded through the 3 additional levels of conformation, and we end up with a functional protein. Or I should say, we can fold it through the 2 additional levels of conformation to get to the tertiary level, and we have a protein at that point. With the additional quaternary level of conformation, it's still a protein, but it's a protein made up of more than one subunit. Now, in order to take that message RNA and translate it into those amino acids, you are going to need a non-membrane bound organelle known as a ribosome. Because we're moving on to RNA transcription, I wanted to remind you again the difference between DNA structure and RNA structure. DNA is double-stranded. It uses the bases adenine, cytosine, guanine, and thymine. And there is only one type of DNA that you find in eukaryotic cells. Now, in contrast, RNA is single-stranded. It uses the bases adenine, cytosine, and guanine, but instead of thymine, you will find the base uracil. And there are multiple types of RNA that you can find in eukaryotic cells. Transcription is similar to DNA replication in that it's done by what I call a transcription machine. This transcription machine is made up of multiple protein subunits that work together. The DNA template is going to be pulled apart into 2 strands, but only one of those 2 strands is going to be used as the actual template for transcription. That template strand is going to be read in the 3' to 5' direction, and an RNA transcript is going to be made so that it grows in the 5' to 3. Now, the nice thing about transcription is all the problems you had in DNA replication are gone. The transcription machine is made up primarily by an enzyme complex called RNA polymerase II. It has helicase function, so it is capable of pulling apart the 2 DNA parental strands. It does not require a primer to bind onto, so it can bind onto the DNA template strand even though it's single-stranded. And probably the most important problem that we get rid of is the polarity issue. The RNA polymerase complex only reads one of the 2 DNA strands. It reads the antisense one that is in the 3' to 5' direction, and it ignores the other strand. And as it moves along that template strand, in the 3 to 5 direction, as all polymerases do, it adds in complementary RNA bases to create an RNA transcript. So where there's a C, it puts in a G RNA nucleotide. Where the DNA base is G, it puts on a C RNA nucleotide. Where the base in the DNA template is a T, an RNA adenine will be added in. But here's the important part. In the DNA template strand, where there is the T base, the complement to that is uracil. In biology, RNA transcription and protein translation is referred to as a process that is known as gene expression. And what is expressed is something called a gene. And a gene is often defined as a transcription unit, which is a stretch of DNA that is transcribed into an RNA transcript. So using the word gene and transcription unit are often done interchangeably. However, I prefer to use the word gene. What is found within a gene is 3 basic parts. You have what is referred to as a 5' UTR. 5' UTR contains the very first nucleotide that you are going to transcribe. It's known as the +1 transcription initiation site. You have what's coming next is known as a coding sequence. In this case, this figure calls it the RNA coding sequence, but you can just call it a coding sequence. And then after the coding sequence, we have what is referred to as a 3' UTR. And in that 3' UTR, there will be several sequences of DNA, one of which will eventually result in the termination of transcription. Now, UTR, we're going to see, stands for untranslated region. It is going to be transcribed. It is not going to be translated. And as we move on in this lecture, you're going to see what these 5' and 3' UTR sections are responsible for. Okay, a transcription unit, or what I like to call a gene, as I said in the previous slide, is comprised of a stretch of DNA called a 5' UTR, a big stretch of DNA called a coding sequence, and then it ends up with a smaller stretch of DNA known as a 3' UTR. Let's start off with what the coding sequence is, because it is the bulk of a gene. The coding sequence is the sequence of DNA that gives rise to the polypeptide strand upon the process known as translation. So the coding sequence is going to code for the amino acids we find in a protein. Not all of the coding sequence is going to become an amino acid upon translation. Stretches of the coding sequence that are actually going to be transcribed and then translated to become amino acids, these are stretches of DNA called exons. And we have we have 4 of them here in this picture. They are the bright green pictures that I'm circling, the bright green segments that I'm circling right now. The in-between regions of junk, which are not going to become amino acids in the polypeptide, these are called introns. And they are spliced out of the coding sequence, so they are not even going to remain in the RNA transcript. Now, the reason why they're called exons and introns is because exons eventually become expressed as amino acids in a polypeptide, and the introns are the in-between regions of junk that are going to get cut out. The UTR regions of DNA, the ones that flank the coding region, these are called untranslated regions. They are regions of DNA upstream, in other words, in front of the coding sequence, and downstream, in other words, behind the coding sequence, that get transcribed into RNA but are not translated into a polypeptide. The UTRs have many regulatory functions in gene expression. For example, they play a role in exporting the mRNA out of the nucleus and into the cytoplasm. They function as protective regions that will prevent the messenger RNA from being degraded. And they have regulatory roles in gene expression, like it could regulate the start of translation. And as I just said, there's a region found just in front of the coding region. That's going to be our 5' UTR, right there. It is found between the promoter and the very first exon in the coding region. So I'll draw this little box and I'll write here E, that's our very first exon. The 5' UTR contains the very first nucleotide that is transcribed. It's often called the +1, and it is referred to as the transcription start site. The 3' UTR is found after the last exon. So I'll put our last exon in here. So it is found downstream of the last exon of the coding sequence. It will contain regions that will terminate transcription, and it even contains regions that will terminate translation. In addition to the gene, we also have additional sequences of DNA that regulate the process of transcription. So I want to tell you about 2 specific regulatory sequences of DNA that are associated with genes, and their job is to regulate the transcription of that gene. The first sequence is called the promoter sequence. It is a sequence of DNA that is recognized and bound by the RNA polymerase. The RNA polymerase is going to be doing the actual transcription. It is capable of recognizing the sequence of the promoter and physically binding to it. And by physically binding to that promoter sequence, the RNA polymerase complex is positioned just in front of the 5' UTR. It is positioned just in front of the +1, the very first nucleotide that it needs to transcribe. Now, this binding of the RNA polymerase complex to the promoter is helped out in eukaryotic cells by small proteins called transcription factors. There are going to be numerous transcription factors that are going to help position the RNA polymerase at the promoter and help it physically bind. There are numerous promoter sequences that have been identified and studied in biology. There are promoter sequences in prokaryotic cells. There are promoter sequences in eukaryotic cells. And when researchers looked at the sequences, they discovered a promoter sequence that was found in both prokaryotes and eukaryotes, and it had the sequence of TATAAT, and they called it the Tata box. The Tata box first evolved in prokaryotic cells, and many of our genes in eukaryotic cells still contain that kind of sequence. The last thing I'll say before we move on to the second regulatory sequence is that in some textbooks, the promoter is considered to be part of a gene. I do not do that. I consider a gene to be a sequence of DNA which is transcribed. The promoter is never transcribed. So by that definition, I don't consider a promoter to be part of a gene, but because it is the key regulatory element that starts and stops transcription, a lot of textbooks consider it to be part of a gene. If you do consider the promoter to be part of the gene, I will not dock you for that. So I won't mark you incorrect. The next regulatory sequence is definitely not part of the gene. It's usually found either a significant number of base pairs in front of a gene. It can be found a significant number of base pairs actually after a gene. It can even be found in the introns of eukaryotic genes. These sequences are called enhancers, and they are short islands of DNA that work together to help enhance transcription. So in this picture here, you can see we have an enhancer sequence. It's made up of a bunch of maybe 6 to 10 base pair little stretches or little islands of DNA that collectively, all of these little short sequences of DNA comprise the enhancer. They are bound by transcription factors called activators, and they actually will physically interact with the RNA polymerase at the promoter to help, you guessed it, enhance transcription. The one last thing you should realize about enhancers is that they are a eukaryotic control sequence. If I remember my molecular biology correctly, most prokaryotic genes have very simple promoters, and I'm pretty sure they don't have enhancers. So they have really, really simple promoters and no enhancers whatsoever. So I generally think of enhancers as being a eukaryotic thing. You'll see lastly in this picture that in order for that enhancer sequence to interact with the promoter sequence, look how the DNA has to actually perform a little U-turn. And that's actually what happens. The enhancers are bent over to be brought into close proximity with the promoter so that all of these promoter sequences and all of these enhancer sequences with their protein helpers can act together to kind of supercharge the RNA polymerase and get it to transcribe that much better. Now, the RNA polymerase needs to be positioned in front of the coding sequence. It needs to be physically positioned right in front of that 5' UTR. That's why the promoter is found in front of the 5' UTR. Think of it as being the docking site for the RNA polymerase. Now, this is true for both prokaryotic cells and for for both prokaryotic genes and eukaryotic genes. Once that RNA polymerase is positioned, it can unwind the 2 strands of DNA, and it will begin to transcribe the antisense strand, which is the one with the 3 to 5 orientation. So it moves down the DNA antisense strand towards the 5' end. Coming out the other end of the RNA polymerase will be a growing RNA transcript. Remember, this RNA transcript is going to be using the base uracil rather than thymine. And once that polymerase clears a section of DNA, the DNA strand reforms its helix. Ultimately, the completed RNA transcript will separate from the RNA polymerase when the polymerase gets to the end of the 3' UTR. So it's gone through the entire coding sequence, it gets to the end of the 3' UTR, and basically the polymerase detaches and the RNA transcript is then released. Now, depending on what gene you've transcribed, we're going to have to modify this RNA transcript so that we can turn it into the kinds of RNA we see in the cell. During the process of transcription, the entire transcription unit or the entire gene will be transcribed by the RNA polymerase complex to form a piece of RNA called an RNA transcript. Now that means the 5' UTR is transcribed, the entire coding sequence is transcribed, both exons and introns, and the 3' UTR is transcribed. But enhancers are not transcribed and the promoter sequence is not transcribed. Only the 5' UTR coding sequence and 3' UTR are transcribed. You'll get a piece of RNA called an RNA transcript, and then, depending on what kind of gene was transcribed, the RNA transcript is going to be processed. It's going to be modified in specific ways to form a specific type of RNA. And there are numerous kinds of RNA in eukaryotic cells. In this course, we're going to look at 3 specific types of RNA. We're going to look at mRNA, tRNA, and rRNA. So following transcription, the RNA transcript of mRNA is going to get processed in a specific way. The RNA transcript for tRNA is going to get processed in a different way. And the RNA transcript for rRNA is going to be processed in yet another different way. So the kinds of genes that give rise to mRNA after transcription, these are our protein coding genes. mRNA stands for messenger RNA, and its job is to carry the genetic information that ultimately will be translated into a polypeptide sequence. The mRNA is easy to recognize in eukaryotic cells because it has at its 5' end something called a methylated cap, which we can see right here. It's actually a modified G nucleotide with a methyl group, and it's attached through 3 phosphate groups onto the 5' UTR. We also have at the 3' end what is called a poly-A tail. I know there's only 3 adenine nucleotides here, but it's actually a string of something like 100 to 200 adenine nucleotides that get added on to the 3' UTR. And then finally, the third defining characteristic is the coding sequence has no introns. Your coding sequence is all exons. In humans, mRNA is transcribed in the nucleoplasm by RNA polymerase II. And the RNA transcript that comes out after that transcription will be modified by adding on a cap, adding on the tail, and getting rid of the introns. rRNA genes are transcribed to form an RNA transcript, which is then processed in a very specific way to form rRNA, or what is known as ribosomal RNA. Ribosomal RNA combines together with several ribosomal proteins to form either the large ribosomal subunit or the small ribosomal subunit. This transcription in humans is done in the nucleolus, so that area of the nucleus called the nucleolus, by RNA polymerase I. The rRNA pieces are going to be assembled together with proteins in the nucleolus into those large and small subunits, and then we're going to export those 2 subunits out into the cytoplasm, where we're going to assemble it to form a functional ribosome, which will participate in protein translation. Now, this picture down on the right-hand side actually shows you what rRNA looks like. It is made up of multiple regions called stems. So I'm going to highlight what is a stem here. These are complementary sequences of RNA, so they actually form double-stranded RNA. And then we have regions of what are called loops. This is single-stranded RNA where there is no complementary sequence, so they don't double back on each other to form a stem. This is a very, kind of looks like a subway system, doesn't it? It's a very complex structure, but this is what a typical rRNA molecule looks like. tRNA genes are going to be transcribed and then the RNA transcript is going to be modified into transfer RNA. The job of transfer RNA is to translate the message coded in our messenger RNA into a polypeptide sequence, so that polypeptide sequence can become a functional protein. In humans, the tRNA genes are transcribed in the nucleoplasm by RNA polymerase III. They are then processed to form this very unique structure, and then they get exported out into the cytoplasm where they're going to perform their job in translation. Now, when we talk about translation, I'll come back to this, but I want to introduce the idea that our tRNA gets folded up into this distinctive T shape. It has 2 very important sites that you're going to learn about. One I'm going to circle right now is called the anti-codon site. That is the site that's actually going to bind onto the messenger RNA. And at this end, we have what's called the amino acid attachment site. And as its name implies, this is where we're going to attach the amino acid. In eukaryotic cells, genes that are known as protein coding genes will be transcribed into an RNA transcript that will be modified to form messenger RNA. So the 5' UTR coding sequence and 3' UTR all will be transcribed to form an RNA transcript that will be made up of the RNA version of that 5' UTR, the RNA version of that coding sequence, and the RNA version of that 3' UTR. Now, it's an RNA transcript. We haven't got mRNA yet. To make mRNA, you have to do 3 things. And I want you to remember this phrase: cap, tail, splice. The first modification is the addition of a 5' methylated cap added to the 5' UTR. And I'm going to circle it. There it is, this little red block right there. It is a modified G RNA nucleotide. It's added on to the very front of the 5' UTR, and it is needed in order for the mRNA to get exported out into the cytoplasm. And it also slows down the degradation of the mRNA by enzymes that exist out in the cytoplasm. Finally, the 5' cap is really important in starting the process we call translation. At the 3' end of the RNA transcript, you will add on a second thing called a poly-A tail. This is a very, very long tail of about 150 to 200 adenine RNA nucleotides. And it is going to be added on to the 3' end of the RNA transcript. Just like the 5' methylated cap, it's needed for export of the mRNA out into the nucleus. It slows down degradation of the mRNA by these enzymes, so it stops the degradation at the 3' end. And it plays a very important role in stopping, or what we call termination of translation. And then finally, the third modification is all introns must be removed from the RNA coding sequence. The coding sequence of an RNA transcript cannot contain introns. Every single intron, which is part of the coding sequence of your RNA transcript, has to be cut out through a process called splicing. Splicing happens in the nucleoplasm, and it's done by a machine called a spliceosome. The spliceosome is made up of several subunits. I believe it's made up of 5 subunits. Each subunit is called a SNRP. A SNRP stands for small nuclear ribonucleoprotein. And what a SNRP is a bunch of proteins and a piece of RNA all connecting them together called snRNA, small nuclear RNA. 5 of these SNRPs are going to come together to form the spliceosome. And splicing is not a random process. It requires specific sequences called splice sites. These splice sites are found where an exon and an intron meet one another, so that the spliceosome knows where the introns are. And during the process of splicing, the spliceosome is going to recognize these splice sites, bind to these splice sites, loop out the intron, kind of form a little lariat structure, cut it out, and then it's going to join the 2 exons together. So in a way, splicing is kind of like editing a film, in which you, the editor, you're going to be the spliceosome, are going to look at the film and you're going to take out and cut out a piece or a scene of that film that you don't want anymore. That's the intron. And you are going to physically join the remaining scenes together. Those are the exons. So splicing results in a final coding sequence in the RNA transcript that is made only of exons. As I've said a couple times, you have to take all your introns out of the coding sequence in the RNA transcript, because these introns are not translated. They do not become amino acids. So all of those introns have to be cut out of the coding sequence in the RNA transcript. But you can also cut out exons. So exons can be spliced out along with introns. And by cutting out exons, you produce new forms of the RNA transcript, new forms of the mRNA. And you can get new types of polypeptides being made. So depending on what exons you cut out and what exons you leave in, you can get an entirely unique combination of exons, which becomes a different polypeptide. This is called alternative splicing. And in this figure, you can see protein A is made up of exons 1, 2, and 3, and protein B is made up of exons 1, 2, and 4. So in protein A, exon 4 was spliced out along with all of the introns. And in protein B, exon 3 was spliced out with all of the introns. This is kind of a space-saving measure if you think about it in eukaryotic cells. We have about 30,000 protein coding genes in our human genome, and yet we make well over 500,000 polypeptides. So how does 30,000 genes correspond to 500,000 polypeptides? Simple. The process called alternative splicing helps create that increased number of polypeptides that we find in our cells. Translation is defined as the process of converting an mRNA message into a polypeptide strand of amino acids. Specifically, it's the process of converting the exons of an mRNA message into a polypeptide strand of amino acids. This polypeptide strand will be processed into a mature functional protein by taking it through the secondary, tertiary levels, and maybe the quaternary levels of conformation. This is performed by the ribosome in combination with tRNA molecules, and the site for translation is the cytoplasm. The exons that we find in the coding sequence of the mRNA is read in groups of 3 nucleotides, which are known as codons. Now, a codon is defined as a group of 3 nucleotides. Codons exist in the DNA coding sequence, they exist in the coding sequence of the RNA transcript, and they exist in the coding sequence of the processed mRNA. Each mRNA codon codes for one of the 20 amino acids that we use to make up proteins. So in other words, translation is going to use something called the genetic code. And what you see here on this picture is what's called a codon table, in which you can see what the codons code for specifically. Most amino acids you can see in this table are coded for by more than one codon. For example, alanine is going to be coded for with the codons GCU, GCC, GCA, GCG. So you're thinking, if the codon has a GC, great, that means it's alanine. Well, not necessarily, because take a look at serine. Two of the serine codons are AGU and AGC. And you're going to say, okay, if you start the codon with an A and a G, that's a serine. But arginine codons are AGA and AGG. So there goes that rule. So you're thinking, wait a minute, it's the third codon that determines the amino acid. There are no rules. Okay? The codon table is the codon table. You just have to look at the codon table and match the codon up with the right amino acid. So there is no rule that associates the sequence of a codon with a specific amino acid. Okay, I lied. There's one rule. AUG is methionine. Other than that, there is no rule. Okay, I lied. UGG is tryptophan. Okay, but those are the only two exceptions. I promise you that when I have you transcribe and translate on an exam, and I will have you transcribe and translate on an exam, I will give you a codon table. And when you are translating these exons into their amino acids, you may write the full amino acid out. You may write the 3-letter abbreviation, or you could write the 1-letter abbreviation. But be aware, on an exam, I'm going to ask you to be specific. I'm going to say, translate this message into an into a polypeptide strand and use the 3-letter abbreviation. This just makes it easier for me to grade it on Canvas. But when you are doing exercises on transcription translation, you can write the amino acids out any way you want. This slide shows us the 3 components of translation. We're going to need transfer RNA, we're going to need a ribosome, and we're going to need the messenger RNA. The tRNA molecule is a single strand of RNA, and it's only about 80 nucleotides long. It gets folded into a very distinct secondary shape, and you can see it kind of looks like a T, and then it's folded into a very characteristic 3-dimensional shape. Let's go back to the 2D shape, because the 2D shape will show at one end a specific region known as the anticodon site. There it is right there. This one has the particular sequence of AAG. This is going to be where the tRNA will physically base pair with the codon of the mRNA template. So in the mRNA, the sequence would be UUC. And that tRNA would temporarily base pair, and I picture it as sort of docking onto the mRNA at that codon. At the other end, there is the amino acid attachment site. This is where the amino acid is going to be found. The amino acid has absolutely nothing to do with the anticodon site. The amino acid that you would find on this particular tRNA is going to be defined by the codon sequence UUC. There are enzymes out in the cytoplasm who will bind this tRNA. They will look at that anticodon site and they will say to themselves, AAG, what's the opposite to that? UUC, up. Now what amino acid codes to UUC? I can't remember. Is it phenylalanine? Go and look at the codon site. That's the amino acid that goes there. It has absolutely nothing to do with the anticodon sequence and everything to do with the codon sequence that tRNA will bind to. Again, what makes this thing? RNA polymerase 3. It will fold into its characteristic shape spontaneously. No energy is required because there's an awful lot of complementary base pairing going on. So these regions in yellow are formed by complementary regions that pair up with each other. And those little loops, they're actually called loops, are regions where there was no complementary base pair. This 2-dimensional shape then folds even more and twists itself into that 3-dimensional shape. The anticodon site is right there. The amino acid would be attached right here. Often the textbooks will use sort of this little peg shape. So the amino acid basically would go right here. And a lot of textbooks will use that as a very simplified 3-dimensional tRNA. The ribosome, again, is made up of 2 subunits called the large subunit and the small subunit. They are created in the nucleolus. The small subunit in eukaryotic cells is called the 40S small subunit. It is a piece of rRNA called the 18S rRNA and 33 proteins. So I was pretty close. I guessed pretty well. The eukaryotic large subunit is called the 60S subunit, and it is made up of, I was close, 46 proteins and 3 pieces of rRNA. S, by the way, stands for Svedberg, which is a unit of sedimentation. So if you are the 60S subunit, you will sediment faster through a gradient when they spin you in a centrifuge versus the 40S subunit. Same thing with these pieces of RNA. Each of them sediment through a gradient when it's spun in a centrifuge at a very specific rate. The rule is the higher the number, the faster it sediments, the bigger the structure is. On the small subunit, there is a site for binding the mRNA. So cell biologists decided to call it the mRNA binding site. On the large subunit are 3 very distinctive regions called the E exit site, the P site, the peptidyl tRNA binding site, and the A site, the amino acyl tRNA binding site. I personally like calling them P and A because peptidyl tRNA and amino acyl tRNA, that's a mouthful. Put it very simply, the P site is where you are going to find the tRNA with the old amino acid attached to it. Actually, at the P site, you'll find the tRNA with the peptide attached to it. And the A site is where you'll find the brand new incoming tRNA with the amino acid that you want to attach. Empty tRNAs leave the ribosome through the exit site. Now this, I'm going to try and describe it to you, and I'm going to confuse the heck out of you. So honestly, I've given you a couple animations to look at, both here and in your Canvas page. You've got to look at these animations because it can be a little confusing when I try and describe what's going on. Just as we saw in transcription, translation has 3 stages: initiation, elongation, and termination. In order to start translation, eukaryotic cells require small protein helpers called eukaryotic initiation factors. Just like in transcription, eukaryotic cells need proteins to help, and they were called transcription factors. In eukaryotes, translation needs little protein helpers, and we call them eukaryotic initiation factors. The reason why they're called eukaryotic initiation factors is that prokaryotes also need help to start translation, and we just call those initiation factors. So the letter E helps us distinguish and helps us learn that we are looking at eukaryotic initiation factors. The actual process of translation is called elongation, and termination is the stopping of translation, and it will happen at one of 3 stop codons. So it will happen at specific sequences that we call stop codons. The last thing I'll say about translation is that it is driven by the hydrolysis of GTP. I know a lot of cellular processes are driven by the breakdown of ATP, but there are some that are driven by the breakdown of GTP, and translation is one of them. So let's take a look at how translation is started. Just like the initiation of transcription, the initiation of translation requires multiple steps. So several things have to happen in a particular order. During the first step, step 1, small ribosomal subunit recognizes the mRNA by the presence of the 5' methylated cap. So it recognizes the 5' methylated cap, and then through this cap, it binds the mRNA sequence at the 5' UTR. The small ribosomal subunit is then going to glide down the mRNA. It's going to move through the 5' UTR towards the 3' end, and it is looking for a particular RNA sequence. It is looking for the very first AUG it can find. That very first AUG in the mRNA sequence is called the start codon, and it codes for an amino acid called methionine. As the small subunit is running through or gliding through the 5' UTR, looking for the first AUG, it is bringing with it the very first tRNA. This very first tRNA is called the initiator tRNA, and it is going to have the amino acid methionine at the other end. So this right here is our initiator tRNA. It is gliding along the 5' UTR because it's being carried by the small ribosomal subunit. The The small ribosomal subunit stops at the very first AUG, and that initiator tRNA is positioned right there where it's supposed to be. All we need to do now is just add our large ribosomal subunit, and the initiation of translation is done. To be honest with you, describing the process of translation is more confusing than just looking at an animation. So there are numerous video animations that you can watch. I'm going to recommend you do that, but I'm going to try and describe to you as simply as I can the process of translation, otherwise known as elongation. The picture we have here on the right-hand side, let's take a look and see what we have. We have our mRNA, we have our small subunit, we have our large subunit, we have the E site, which is empty. We have the P site, which contains a tRNA linked to 4 amino acids. So this P site tRNA is linked to the growing polypeptide. And coming in now to the A site is a brand new amino acid with a brand new tRNA with a brand new amino acid. That tRNA with the new amino acid you want to add on to the polypeptide slots into the A site. Now what happens is the ribosome is going to take this polypeptide off of the P site tRNA, and it's going to move it over to the A site tRNA, and you're going to form a peptide bond. The ribosome now is going to head to the 3' end of the mRNA, and when it does that, the P site tRNA, which has lost its polypeptide and is now empty, moves into the E site and exits. A site tRNA, which has now got the polypeptide attached to it, slots into the P site, and then the A site is empty and ready to receive a brand new tRNA with the newest amino acid you want to add to the growing polypeptide chain. And the cycle is going to repeat over and over and over again. The elongation process stops when you get to 3 very specific codons. They are called the stop codons. They are UAA, UGA, and UAG. When the ribosome reaches these sequences, in other words, when these sequences enter into the A site, there is no tRNA that will come in. No more amino acids will be added. Instead, what slots into the A site is something called a release factor. And when that release factor slots in, the ribosome basically comes apart into its subunits, and the polypeptide is freed. Now, before we leave this slide, I'm going to tell you something really, really important. When you are translating these either on practice exams or quizzes or even on your actual exam that I will give you, if you write start when you see the AUG, the very first AUG, I will take a half a point off. There is no such amino acid as start. I don't care what codon tables do. Codon tables write start, it drives me insane. Because in that coding sequence, there are other AUGs. What are you going to write? Are you going to write start every time you see an AUG codon? No. Every AUG codon you will see in that coding sequence, you will write methionine, even the first one. When you get to the codons UAA, UGA, or UAG, you will not write stop. I don't care that the codon tables say stop. There is no such amino acid as stop. What you are going to do when you get to those 3 codons is you are going to stop writing. Put your pen down, step away from the keyboard, drop the pencil, whatever you have to do. Don't write anything. That's going to be impossible. Your hands are going to shake. You're going to start to sweat. You're going to need to write something. Okay, you may put a little asterisk, and that will, to me, denote the release factor. But if you put stop, I'm taking a half a point off, which means on an exam, if I have you translate a piece of mRNA and you write start and a bunch of amino acids and stop, you have now lost full points for that question. So don't lose points for this by writing start and stop. Write methionine, and then if you have to put something down, put a little star or a little asterisk. Don't write stop and then cross it out and go, oh, we're not supposed to write stop, professor. And I'm like, yeah, you're right. You're not supposed to write stop, -1/2. Okay, so don't get cute. Here's a really nice summary of how translation takes place. The bottom part is what I want you to pay attention to. I want you guys to note that just because one ribosome is on the mRNA doesn't mean it's the only ribosome. The minute a ribosome assembles at the 5' end of the mRNA and starts to move to the 3' end, as soon as there's room, another ribosome will assemble. As soon as that one gets out of the way, another one will assemble. So each mRNA strand can actually have multiple RNAs all moving along, translating them kind of like a conveyor belt system. Translation is a great example of a cellular process that really is simple once you watch an animation. It sounds really confusing, but honestly, once you see the animation, it's not that bad at all. This YouTube video that I have here is probably my favorite. It's a company called Virtual Cell, and it does a fantastic job of showing you what happens during translation. Check it out. And also, I want you to check out a few other translation animations. Just like you did with transcription, go to YouTube, type in translation animation. You'll get a whole bunch of them. Many of these translation animations are combined with transcription. Some of them will go through the entire process of DNA replication, transcription, and translation. I would recommend you check out several of these animations so you get the overall view of gene expression, which is transcription and translation, and then how DNA replication and gene expression are related to one another.